years, and then talk about some of the research we're doing here at Inova to better understand that. I don't have anything to disclose financially. But when we talk about head and neck cancer, I often have to explain this to my kids. We're not talking about brain cancer and others. We're talking about cancers really of the upper aerodigestive tract. So the nasopharynx, the oral cavity, oropharynx, larynx, and hypopharynx, and also the salivary glands and sinuses, which can give rise to cancers. When we think about HPV head and neck cancer, most of them occur in the oropharynx. So back in the back of the mouth, the tonsil and base of tongue area, there's a specific lymphoepithelium cells that line that component of the upper aerodigestive tract where these cancers seem to arise. Although we also are starting to see HPV tumors that seem to express HPV markers in oral cavity and larynx cancers, which we're still trying to make sense of. If you go to the next slide, head and neck cancer, as I mentioned, is about 95% are in the aerodigestive tract. Most of those are squamous cell carcinomas. And then in the salivary glands and paranasal sinuses, there are a range of other subtypes, histological subtypes that we can see under the microscope. There's actually 25 different kinds of salivary gland cancers that all behave a little differently, although they are very rare as a group. And then we can also see other cancers in the head neck region. Certainly melanomas can occur in the head neck area. Lymphomas can present in the neck. Rhabdomyosarcoma is a cancer mainly in pediatric populations, but can happen to adults, can happen in the head neck region. But most of the time when we're talking about head and neck cancer, it's squamous cell carcinomas of the nasopharynx, oral cavity, oropharynx, hypopharynx, and larynx. If you go to the next slide, it's not a rare cancer, as you all just answered. It's actually, historically, the Commission on Cancer and state-based tumor registries list oral cavity and pharynx cancers separate from larynx cancers. But if you add those together, as of 2020, 48,000 men would be diagnosed with head and neck cancer last year, and less. But there's it's about the 12th most common cancer in women, the 6th most common cancer in men. So while it's not one of the big four, it's certainly not rare. And fortunately, because of that, we've learned to better understand how to treat it and have optimized that treatment over the last several decades. If you go to the next slide, the way we treat head and neck cancer is with most solid tumors based on the stage. And actually, we now have a different staging system for HPV positive cancers because they have such a good prognosis, which I'll talk about in a second. But if we think about early stage cancers, stage 1 cancers, oftentimes those are just primary tumors that are small to medium sized, not large, not invasive to other structures, and not involving lymph nodes of the head neck region. Those patients typically are just treated with surgery alone or radiation alone, depending on where the tumors are. There's typically not a role for chemotherapy in those patients. I'm a medical oncologist. I give chemotherapy for head and neck cancer. And the cure rate is exceedingly high, about over 90%. Now, oftentimes, unfortunately, while patients can detect or vigilant primary care doctors or dentists can detect a early stage cavity in the lips or oral cavity, oftentimes these tumors arise in the oropharynx or further back, which patients can't see and oftentimes don't give clear symptoms. So oftentimes we diagnose patients at a higher stage. If patients are metastatic, most commonly this cancer can spread to the lungs. The second most common is the liver. Interestingly, it doesn't go north, it doesn't go to the brain, but in those cases, unfortunately, it's a terminal diagnosis if it has spread. And for those patients to try to help them live better and longer, and that's what we mean by palliative chemotherapy, we're helping them live longer, but also have an enhanced quality of life. We can use chemotherapy to treat those patients, but unfortunately, even with chemotherapy and these days immunotherapy, the average life expectancy with someone with metastatic or recurrent outside the head and neck region, head and neck cancer, is only 12 to 15 months. Now, fortunately, that's less than 5% of patients when they initially present with head and neck cancer have metastatic disease at the time of diagnosis. The majority have what we call locally advanced. So either they have a large primary tumor and or they have lymph node involvement in the neck. And for those patients, there's sort of two paradigms of treating, either surgery followed by radiation. And some of those patients, if they have high risk disease seen under the microscope from the pathology done after surgery, they will need chemotherapy. And also some patients are treated primarily with radiation. In those cases, we definitely give chemotherapy during radiation and hold off on offering surgery only till later if it's needed. There's some nuances to this. For example, oral cavity cancers, cancers of the anterior tongue, buccal mucosa, gingiva, those patients typically get surgery first, followed by radiation with chemo if needed. And some patients if they have larynx cancer, where the surgery would be a laryngectomy, a removal of their vocal cords, for those patients, we typically offer radiation first with chemotherapy. For oropharynx, and again, that's where HPV positive tumors occur, it's sort of an either or. And it often depends on who met the patient first and whether the surgeon is adept at doing what we call transoral surgery, where we have now robotic surgery where surgeons can get to the oropharynx, to the tonsil and base of tongue safely without having to go in from the side, which is much more intrusive with much more morbidity. So nowadays, patients can be offered surgery primarily, even if they have, and that includes, again, a primary tumor resection followed by a neck dissection to take out their lymph nodes. And those patients, if they do have locally advanced disease, again, would get radiation and may get chemotherapy. For the cure rates of those diseases with locally advanced disease, it's still high. It's still a majority of patients that we can cure with that combined modality or multimodality approach. So if you go to the next slide, we think about the risk factors for getting traditional head and neck cancer. Certainly tobacco is the highest risk factor of chewing tobacco, smoking cigarettes, smoking cigars, all that gives rise to risks of getting head and neck cancer. Alcohol, especially heavy alcohol exposure, especially in the setting of someone who also chews or smokes tobacco, is a high risk feature. Interesting, in India and South Asia, there's a post-prandial, post-meal nut that many people chew called betel nuts, and that's highly carcinogenic and give rise to oral cavity cancer in that area. Epstein-Barr virus can give rise not only to mononucleosis, but to nasopharynx cancer, and that's endemic in Asia and China. And a lot of our trials on how to treat nasopharynx cancer actually comes from China, based on that being a very common cancer in that region, especially because of endemic EBV infection. But as we're seeing now more over the last 15 to 18 years, a growing cause of head and neck cancer, especially in the oropharynx, is HPV. So if you go to the next slide, all of you are probably familiar with some of these details. But it gives rise in up to 40% of head and neck cancers. It's in the tonsil base of tongue that I mentioned. It's most commonly from HPV 16. That's 95% of all cases are HPV 16, but we can also see HPV 18, 31, 35, and even some of the rare oncogenic serotypes of HPV 16. Typically, these patients are non-smokers, although if they do have a smoking history, that can impact their prognosis, somewhat diminish their prognosis from otherwise a very curative cancer. And I would say after 18 years, we have studies of looking at head and neck cancer in terms of risk factors. We have conflicting associations looking at exposures and how this epidemic is given rise to. There's been obviously a lot of extensive epidemiology study looking at sexual habits, number of sexual partners, age of sexual activity onset, and again, mainly in males. And while there's been some positive studies in those areas, oftentimes been negative studies looking at that same risk factor. Habits of kissing, marijuana exposure was one that initially was positive, then that now has been disproved. So we don't know who these patients are, and I think we have an incomplete understanding of the transmissibility of HPV infection. But there is growing evidence, I'll talk about it at some length, of that delayed clearance of infection in people who are exposed, again, in ways that we don't completely understand, may be those that give rise to cancer years and even decades down the road. So I'll talk about that more in a bit. So if you go to the next slide, we do know that HPV positive tumors are different than HPV negative tumors. Again, those that are caused by tobacco seem to have a different behavior and a different mutational study. This was the first of many studies done over the last few years, looking at tumors taken out of patients and then tested for genomic mutations in those tumors to see what genes are mutated that seem to be driving that tumor cell to grow, to become a tumor, to become a detectable cancer that needs treatment. One of the genes that drives most cancers actually across the board is in a gene called P53. That's sort of the master cell cycle control gene in all of our cells. Those are highly mutated in HPV negative tumors. As you can see in this case series, all the tumors to the right that are HPV negative have mutations in P53 as well as other genes, where that was only found in one HPV positive tumor seen on the left of this screen. And as you can also see, there's not actually just one gene that's mutated in the HPV positive tumors on the left. Many genes can be involved in driving that cancer to grow. And what's interesting is we definitely see that heterogeneity in terms of mutational profiles. It's not just one gene that's mutated by HPV. There can be many, depending on where the HPV DNA gets inserted into the normal cell, causing a mutation, causing that cell to then grow and become cancerous, which is by definition a mutation that leads a cell to grow when it shouldn't and not to die when it should. So we know that these tumors are different than the traditional HPV negative tumors. And if you go to the next slide, the other key thing that we know so far over the last 10 years is that patients have a much better prognosis. They have much higher cure rates if they undergo treatment. And these are two large studies that came out 10 and 11 years ago. Not to get into the details, but these were large trials on the left asking a question about different uses of radiation, whether to give 5 treatments a week or 6 treatments a week. On the right, whether patients got chemotherapy before radiation and then chemotherapy during radiation. And what these Kaplan-Meier survival curves are showing is what % of the patients are alive 1, 2, 3, 4, 5 years later, or 12, 24, 36, 48 months later. As you can see, the HPV positive tumors have significantly higher cure rates, 20, 30, 40% higher cure rates than HPV negative tumors. So that's the good news, is that patients have a better prognosis. The bad news is it's happening at a higher rate. The key question we're asking clinically across the country in the US and in Europe is, if they have such a higher cure rate, can we de-escalate treatment? Can we give patients less radiation? Can we avoid giving patients chemotherapy at all? Can we substitute immune therapy instead of chemotherapy, which is the new kid on the block, and maintain those high cure rates? So we have a number of large phase 3 trials going on in the US right now asking that question, because for head and neck cancer, while we can cure the majority of patients, we oftentimes leave them with chronic long-term side effects. Their saliva glands get damaged from the radiation, and therefore they have chronic dry mouth. Their taste can be affected. So they go through their years and decades of life after their treatment with diminished taste, and some patients no taste at all. And if you can imagine a lot of what it means to be a human and social, and certainly pre and post-COVID, how we enjoy life is going out to restaurants, sharing meals with families and the rest. And our patients oftentimes can have that significantly disrupted. So it's a high impact in quality of life for our cured patients. So the question is, can we give less radiation? Can we avoid chemotherapy, which also has its own toxicities, and maintain those high cure rates? We don't know that yet today, but that's what those studies are investigating. Can we de-escalate treatment and maintain that high cure rate? So more to come in the years ahead on that question. If you go to the next slide, when we look at this epidemic, and again, it first started being recognized in the early 2000s, but in looking back, there's been some great researchers looking at this, both at the NCI and centers around the country. One of the leaders in this field is Maura Gillison, who's now at MD Anderson. What's interesting is that it seems to be, again, so these curves are birth cohorts broken down by 5-year age groups. On the right is women, on the left is men. As you can see, we're not seeing an increasing rate of this cancer in women based on any age cohort. That's on the right. If you look on the left, at the younger men in their 20s, 30s, into early 40s, we're not seeing a rising increase in those age groups, depending on when they were born. Again, looking at the SEER database from 1970 to 2010. But what we're seeing is that men of middle age and older age is where we're seeing this incredible rise, mainly in their men in their 60s and 70s. But as you can see, a dramatic increase in terms of the rate per 100,000 man years is where we're seeing this growing epidemic. And that's what I would say in my clinic, where the majority of my new patients are HPV positive patients. They're men, and they're men in their 50s, 60s, and 70s typically right now. If you go to the next slide, there was data when we started seeing this growing increase. A group at NCI sort of extrapolated, what do we expect to see over the next 2 decades, and we're definitely seeing this 10 years later, which is a decreasing incidence of larynx cancer. Larynx cancer historically was caused by smoking. That rate was going down dramatically. And oral pharynx, which again is where the HPV positive is, going up dramatically. So even though the tobacco use has been declining in the US since the 1960s and 70s, the number of head and neck cancer patients has actually gone up 50% over the last 10 years, again, driven by HPV and specifically in the oral pharynx area. So if you go to the next slide. So when we've looked at what are the causes for this, again, epidemiology studies have shown conflicting data about sexual habits and other potential co-exposures that might lead to this. There has been brewing data, and this is from a meta-analysis that came out last year, that looked at a whole variety of studies going back to the 2000s and then especially in the 2010s, that looked at if patients were known to have an HPV infection, what was their rate of getting head and neck cancer? In the oral cavity, there is an increased rate, but especially in the oral pharynx cancer, there is a almost 17 odds ratio increase, 17-fold increase in your risk of getting oral pharynx cancer if you had had HPV positive detected in your saliva. And if you go to the next slide, this was from this same study. In the next slide, I'm sorry. So and then in a different study, a different study done. This was done by Maura Gillis and published about 9 years ago. They looked at could they detect HPV saliva infections in men, and this was a large study, 50,000 men, part of a public health study called the NHANES study that's done every year. In the 2010 study, they got saliva samples from subjects in this epidemiology public health study to look for HPV infection. And they found a surprising percent of people infected with HPV, which definitely correlated with age as people got older, with up to 11% of subjects in their 50s and 60s having HPV detected in their saliva. Unexpected finding, and really brought on by this identification of the cancer rate going up, but also our ability to do next generation sequencing and genomic testing to be able to look at the microbiome of the oral cavity of saliva and being able to detect HPV out of that milieu. Technology got us to the point where we could do this study. But surprisingly, we found that up to 11% of people had HPV infection, as you can see in the blue box there, primarily in men, not only in men. 3% of women had HPV infection detected in their saliva, but 10% of men did. So a high rate, 1 in 10 males had HPV infection in their saliva. And as we saw from the previous meta-analysis, that seems to be the risk factor of having an infection and especially potentially a chronic infection giving rise to this. If you go to the next slide, there's been conflicting data about whether race, ethnicity has a role. That previous slide showed no correlation between race and ethnicity in that U.S. population. This is a summary that was published 2 years ago that looked at studies that have been done around the world and what rate of infections they found in those natural history studies, depending on where people lived. And as you can see, the size of the box is based on the size of the study. The color of the box is what percent of people were positive. And as you can see, higher rates in the US and actually in Central America, higher rates in some countries, especially in Europe, but lower rates elsewhere. Conflicting studies in Brazil, low rates seen in Japan and China. So that question of who is at risk, seemingly having a geographic distribution variable, raised the question, is there something about genetics, which is what race and ethnicity at the end of the day are based on, giving rise to this risk of infection? So that was the question that we wanted to answer here at Inova with a pretty diverse patient population. If you go to the next slide, we started in 2010 a Translational Medicine Research Institute. We recruited the former head of NCI, John Niederhuber, to found this institute. It was a large research institute funded by Inova to do genomic research. We had clinical research staff, laboratory staff, as well as bioinformaticians focusing on genomic sources of illness and disease. If you go to the next slide, one of our largest studies that we did was I'm sorry, go to the next slide. Next slide too, all the way through, if you don't mind, was to look at genomic inheritance of illness. Our largest study was what we called our childhood longitudinal study. We wanted to look at newborns. Since over 12,000 babies are born every year at Fairfax Hospital, we thought we could have a large population to investigate questions of childhood illnesses and even illnesses of pregnancy, including preterm birth. And then we looked initially at the first 2 years of children's lives, and now we've extended that up to 6 years. And we looked for components of their parents, the mom, dad, baby, that trio, and looking at genetics and inheritance. We collected a lot of biobanking materials to do these studies, including saliva from both mom and dads. And that served as the basis for and we asked the participants in this research study for broad permission to do investigations into human disease, and including with an IRB approval, looking at HPV infection in these young parents. So these primarily were parents in their 20s and 30s, some in their 40s. And we looked at the question, could we find HPV in these young people's saliva? And did that help give us answers to this question? If you go to the next slide, interestingly enough, in Northern Virginia, we have an incredibly diverse population of patients, and in this case, newborn parents. We had over 110 countries represented in these parents. Not that their ancestors were born in 110 countries, they themselves were born in 110 different countries. The majority were in the US, but significant numbers from around the world. And actually, for a while, we had the largest whole genome collection of worldwide population in the world, literally, as a research study. And we still have the largest collection from people from Central and South America in our studies here, looking at questions of health and disease. But you can look at not only country of origin, but also what we call ancestry. And I'll talk about that in a second. So if you go to the next slide, what we did with this HPV project is we wanted to look at subjects' parents, and in this case, the fathers, the male parents, to see if we could detect HPV saliva in their saliva. We looked at subjects who also had their whole genome sequenced, so we could look for correlations between genomic variants and HPV infection. We were able to extract, and I won't bore you with the details in the lab of how we did the detecting of the HPV genome in their saliva, but we ran a lot of development of that study over a period of about 2 years to have it be highly sensitive and specific to HPV. If you go to the next slide, again, this was our PCR approach that we did, both looking at a component, the L1 version, which defines most of the HPV subtypes, including 16. And then we ran that with both whole genome sequencing and then Sanger sequencing to confirm. If you go to the next slide, that this was our workflow. Again, this is more interesting to lab-based folks probably than the people on the call today. But this was how we did our analysis to look at, in a highly specific and sensitive way, HPV subtypes in these saliva. If you go to the next slide, these were our results from 1,300 saliva samples. We found initially 178, which were positive, and then that was confirmed to be 82 men in this studies. 68 we confirmed by Sanger sequencing. 14 patients we had questionable results, and 6 of those patients those equivocal results because they had multiple HPV subtypes in their saliva. In total, we found 28 different subtypes. If you go to the next slide, these were the different types of HPV that we found. HPV 13 was actually the most common, but close behind was HPV 16. Again, that's the oncogenic version of the most oncogenic version of HPV is HPV 16. But we also found patients who had other serotypes, 18, 31, and other high-risk versions of HPV in these saliva. So then we asked, what was the correlates of having this infection? If you go to the next slide, what we wanted to, and then we had not only these 82 cases, but we had a large collection of negative controls, almost 1,200 fathers who we had negative controls to be able to ask questions who seemed to be at risk for having HPV infection. If you go to the next slide, and this is just our whole genome sequencing methodology that, again, I won't bore you with, that described how we looked for variants of interest and also defined ancestry. So if you go to the next slide, a key thing when you look at whole genome sequencing and you're trying to tease out differences amongst in a population, you want to make sure none of your subjects are related. There were no father, sons, or brothers in this cohort, and we did not find any first-degree relatives. So that was a key first data clarifying point. If you go to the next slide, then we defined, and again, this is well established in the whole genomic literature, ancestries. And when we say ancestry, interpret that as being ethnicity or race by as defined, again, over the last 10 years in the whole genome research field. And how we define different ancestries is, in this case, is again 9 different groupings: African, African American, East Asian, European, Hispanic 1, which is basically Central Americans, Hispanic 2 is South Americans, and then South Asians, and then Pacific, and then Asian and Pacific Islanders or other. And this is just the scatter plot of the frequency of those fathers and what ancestry that they had based on that, again, whole genomic description of who has what ancestry. Again, read that ethnicity or race. Next slide. And this is what we found in terms of who had HPV infection. So, and this starts to get at, is there a genomic risk for having this? And interestingly enough, we found different incidence rates of this. While small numbers, the rate in Africans and African Americans was actually very high. We also found high rates in South American Hispanics, but actually very low rates, actually 0 rates in East Asians and low rates in South Asians, which sort of confirms what you saw on that map a few slides before. So definitely a statistically significant difference in terms of the risk of having HPV detected in this random one sample collection based on ancestry, based on genomically defined ancestry. So interesting. So we want to keep going with this research. And if you go to the next slide, we wanted to see, did it matter by age? As I described to you earlier, in Mora's study, they did see an increasing risk in age. And we want to look at our population here. As you can see, the red curve is those that are positive HPV. The blue curve is the HPV negative patients in our study. We did see a slightly older population in the fathers who had HPV detected in their saliva by about 1.5 years. There had been some studies that showed that obesity was a risk for HPV infection. We did not see a difference, statistically significant difference based on body mass index in between the positive subjects and the controls. Another question, if you go to the next slide, was, does smoking, is that a risk for having HPV infection? Because as I told you, it impacts prognosis if you have cancer, but did it impact the rate of having this? And we did not see a statistically significant difference between non-smokers and smokers in terms of HPV infection in this cohort. And then if you go to the next slide, the sort of key thing that we were looking for is there a genomic variant that may, or variants that may predispose people to having chronic infection? And what this is what's called a forest plot, looking at individual genomic variants, SNPs, single nucleotide polymorphisms in the genome. So a variant between subjects that can be, normal or disease causing. And so the graph on the left is showing the different chromosomes. So we have 22 plus our gender defining chromosomes, and then there are variants that we tested in this, again over 300,000 variants in all of the chromosomes. And what surprised us, and what you love to be able to see when you do whole genomic analysis for a risk or a causative factor, is was there a single variant that seemed to jump out at you? And as you can see, especially in the higher dot there, but also both dots there are on chromosome 3, in actually an intron area of the genome. At that point, we are now seeing 2 variants that seem to predispose these individuals in this relatively small study to have HPV infection in their saliva. So very intriguing, unexpected, and now something that we're diving into in much more greater depth with both here at Inova and at the NIH to try to understand how this might explain this risk factor. So a hypothesis causing and early finding, but one that we're excited to look at. Now, if you go to the next slide, our next steps is trying to understand those SNPs, what they might do functionally. Again, when you think about our DNA and our genome, There are coding regions of the genome that code for proteins. Those are called exomes. This was actually found in a non-coding region. So it's not a mutation or a polymorphism in a gene that we know of that's involved in immunosurveillance or our immune system or our epithelial cells. So we don't, so again, it's not obvious how this variant, these two variants might be explaining this risk. So that's why we got a lot of work to do from a research and a back to the lab kind of question. But our next steps are to look for this also in a similar matched female study. So we're investigating the moms from the study to see if they have the rate. Again, we might expect a up to 3% rate in these moms of having HPV in their saliva. We'll look at correlations between spouses, and we'll also look at correlations genomically in those subjects. And then we're looking and collaborating with some other investigators, again, including Maura Gillison at MD Anderson, to try to look for confirmation of our findings, because that would be, from a validity standpoint, that's what we're looking for next, to try to see if our findings were not just a fluke, but were actually mean something. So that's our research that we've been doing here at Inova that's ongoing. If you go to the next slide, what to do about this in general is, as we all know, is vaccination. And you all know, and VHIT has a priority, especially vaccinating adolescents. But as we all know, it's approved to prevent Gardasil, and now Gardasil 9 is the only vaccine now in the U.S. approved to prevent cervical cancer, but as of last year, also approved by the FDA to prevent head and neck cancer. By the math, we expect more HPV positive head and neck cancers to happen this year than cervical cancers. Not to say it's either or, but it's both cancers and anal cancer, as we all know, that we can prevent with a vaccine. And now that it's approved up in men and women up to the age of 45, it's not just pediatricians that need to be focusing on this. It's primary care, it's internal medicine, family medicine, and dentists that need to be aware of this and counseling their patients about this availability. Because it's one of the only times we have an anti-cancer vaccine. And as you can see from my earlier slides, while we cure most patients with this, we don't cure them all. 20% of my patients that I see with HPV positive head and neck cancer will succumb to their disease by its recurrence and metastases, and will die of that cancer. And that's thousands of patients a year across the US and more across the world that we could prevent if we had more use of the vaccine across all age groups, at least in those age groups that it's approved for. So if you go to the next slide, what can we all do, especially the providers, the peers on this call, is be looking for this disease, given its commonality. So certainly on an oral exam, oftentimes if the dentist or primary care physician sees an abnormal tonsil in an adult that warrants further investigation, certainly going into an ENT is ear, nose, and throat or otolaryngologist for evaluation is important. And oral surgeons are involved in treating this disease, especially in the oral cavity, less so in the oral pharynx, and they don't typically do neck dissection. So typically an oral surgeon can get us a diagnosis, but not the definitive surgical treatment of the disease. That needs to be done by an ENT surgeon, and ideally one who's trained in head and neck cancer surgery. That's an added fellowship that ENTs go through, and ideally someone who does a lot of cases a year, at least 10 or 20 cases a year. We know the quality improves by a surgeon if they see enough of this on a yearly basis to be good at it. If the initial finding is in a lymph node, again, seen by a primary care, family medicine, or a dentist, certainly, and oftentimes you can't see anything on oral examination, the first steps could be treating it with antibiotics. Certainly upper respiratory infections can present with abnormal lymphadenopathy in the neck. So a run of antibiotics certainly is appropriate, but close follow-up is needed. And if it doesn't resolve that enlarged lymph nodes, an ultrasound-guided fine needle biopsy is the next step. We don't want to have a surgeon take out a whole lymph node. That's the old way of thinking about lymphoma. We don't even need that for lymphoma these days. And for head and neck cancer, if a surgeon takes out a full lymph node, there's a risk of tumor spill into that tumor bed, which can affect the prognosis of that patient, actually, if they undergo a lymph node, isolated lymph node resection for a diagnosis. The next step is a CT of the neck to evaluate a primary source and extent of the lymphadenopathy in the neck, and then a referral to an otolaryngologist for further evaluation, biopsy, and then treatment planning. Again, a common disease that we all can be on the lookout for In the US, the average length of time between, and again, typically a male, between them noticing something, a symptom, sore throat, problem swallowing, or a lump in their neck, to getting diagnosed with head and neck cancer is 9 months. Now, it's probably been brewing for years, if not decades, but that 9 months certainly doesn't help from a prognosis or cure rate perspective. So if family medicine internists treat presumed tonsillitis or pharyngitis that's presenting with lymphadenopathy and a sore throat with antibiotics, you can't ask that patient to come back in a month or two if it doesn't get better, because sometimes it can improve a little bit, but not resolve completely. Close follow-up for those patients. And again, we don't know who those are at risk for. It's not smokers that you're worrying about. You're worrying about everybody for that. So close follow-up is required to make sure that there's not a cancer brewing under that lymphadenopathy that is seen, and close follow-up after antibiotic usage. If you go to the next slide, just want to shout out Erica and the work being done by the Virginia Health Catalyst. This is, I think, going to dramatically improve not only the care of our residents in Virginia, but also increase the care in head and neck cancer treatment. If you go to the next slide, as you know, currently Medicaid does not cover adults. It covers children and pregnant people for comprehensive dental care, but only emergency extractions in adults. And as of July 1st, largely to the credit of the Virginia Health Catalyst, the American Heart Society, and other advocacy groups in Virginia, if you go to the next slide, we now will have comprehensive care in adults 21 years and over. That's going to affect over 800,000 Virginians that have Medicaid. And for our head and neck cancer patients, again, for screening, for detection of oral cavity lesions, oral pharynx lesions, and also a key part of our patients who do go through this treatment, oftentimes they need dental work before they start radiation. If they have loose teeth, if they have roots that need to be addressed, we'll have patients get extractions done before radiation, and then they can follow up a year after radiation to get implants or crowns placed. Oftentimes that's a significant delay in patients who have Medicaid. We can take care of their cancer treatment, but we need a dentist to help us with that dental work prior to starting radiation. And this is just going to be a huge help in terms of getting that work done in a timely way so that their care doesn't get delayed and their prognosis does not worsen from that diagnosis. If you go to the next slide, this is a summary of the care that'll be provided by this benefit. The bottom line, if you go to the next slide, shows that basically there's no annual limit for adults, up to 3 cleanings per year, including dentures and anesthesiology. So a very comprehensive benefit going live July 1st. Again, that Eric, as you know, has been talking about to our group here and other groups around the state to make sure that dentists and primary care physicians are aware of this. So if you go to the next slide, more information on their website, on Erica's website to look at that. So important new care availability to our patients starting this summer. If you go to the next slide, I don't want to belabor this too much, but since some of you around the state might not have heard of Inova and our cancer program, we've been working on our cancer program over the last 7 years. Our new cancer center opened up 2 years ago, very much like an academic model where we have disease specialists focusing on their disease areas. All I treat is head and neck cancer. We have specialists across the different diseases for adult and pediatric malignancies. With a treatment approach, treating our whole patient, high focus on clinical research and patient-centered care. If you go to the next slide, we are now the largest group in the DC region in the Northern Virginia with a wide variety of specialists. If you go to the next slide, if it's not advancing, there we go. Wide specialists taking care of a range of patients and malignancies. And if you're interested in learning more, if you go to the next slide, there's more information at our website. There's even a virtual tour on YouTube, and we have a line for physicians as well as for patients. So that's and the last slide is just to say thank you for allowing me to participate in this peer-to-peer series, and thank you. I'd be happy to answer your questions. And actually, the first one that popped up, can you tell us more about the route of transmissions? Can males give HPV to females by kissing? So the answer is we don't know. When studies have looked at spousal transmissibility, we have not seen a higher risk in females getting this if their male partners or spouses had HPV detection in their tumors. There's a given that it's mainly a male disease, we don't know why that is. We certainly, there also studies have not shown a higher risk in men whose female partners have a history of abnormal pap smears. So again, it seems to be more readily transmitted. And again, there's studies going both ways, but there's not a obvious, from my reading of the data, an obvious one way of this virus being transmitted. It's certainly probably from close contact. It probably is not from fomites, from silverware and touching doorknobs and the things that we used to be worried about with COVID. But in terms of its likelihood of transmissibility and the timeline from infection to cancer is still being defined. It's probably years to decades. So the exposure to a head and neck cancer patient, he was a 55-year-old male, could have come 20, 25 years prior. But certainly there was one study in France that looked at deep kissing and looked at that transmissibility. That study was positive, that showed that extensive and deep kissing was a risk factor between men and women, but other studies have not confirmed that. So I would say at this day, at least what I tell my patients is, we don't exactly know how it's transmitted. It's certainly probably close human contact is where it is. HPV is mainly, obviously, as you know, the reservoir is humans. It's not transmitted from animals. But in terms of exact risk factors, exact habits that may help us separate patients who are at high risk and low risk, I don't think there's data yet to show that. So therefore, we need to be screening everybody and offering vaccines to everybody, especially those under 45. In my clinic, if a patient and a spouse are unvaccinated, and most adults over 50 are not, if it's a female patient, I encourage their male spouse to talk to their primary care doctor about getting the vaccine. There might be there hasn't been a study looking at the male spouses of female patients with head and neck cancer, HPV positive head and neck cancer. There's a theoretical, given how predisposed males are to having this, I think there's a concern there. For studies that we did when I was at Georgetown and MD Anderson did, we didn't see a higher rate in female spouses of male patients. So in those, I certainly encourage them to talk to their primary care doctor about the vaccine. As you know, if for people over 45, insurance doesn't necessarily cover it. It's about a $600 vaccination series for adults to pay for on their own, which certainly has a financial toxicity to it. But I encourage them to talk to their primary care doctor about that, especially the male spouses of my female patients. But again, transmissibility is one we still have to better understand. As we're finding out in these studies, the exposure might happen early in life. And beyond that, I don't know if we have enough knowledge to either say how a patient got it or how to triage patients into risk patterns based on those previous exposures in their life. So it's one we all have to be on the lookout, unfortunately. But good question.
Feel free to unmute yourself or put your question in the chat. While we do that, Dr. Deeken, in the results that you had about the breakdown, was there anything that could potentially point to cultural or understanding of HPV or even access to healthcare as to why certain cultures had greater prevalence or incidence?
Yeah, great question. And again, these were all typically, first generation immigrants to the U.S., but people from other countries. So that, the one thing we couldn't control for is what was the rates before they got here. So we know that these are all adults who've lived in the U.S. for some period of time, 1 to 30 years. So it's controlled for that, but so we can't really take back to them what their risk is to their countries. But we did see, interestingly, certainly an ethnic, racial, country of origin difference, which is what led us to the genomic question. So, and because we don't know what to do with this data, we certainly didn't tell those patients, we don't have the data yet to say, is that you should get the vaccine because we detected one saliva sample that was positive. There's a growing literature, I would say, over the last 2-3 years looking at chronic infection. And when they talk about HPV chronic infection, normal clearance is thought to occur, again, based on different studies looking at this in different ways, anywhere from 3 to 18 months can be a clearance. If you had an exposure that led to an infection, how long does it take your body to clear? And then we talk about those that have prolonged failure to clear. So over 12 months, certainly over 18 months. And again, those studies are just going on. There's a large HPV study that I'm sure you all know, looking at people in the US, Mexico, and Brazil. It's called the Human HPV Study. And over time, I think we're looking at what those clearance rates are. So, today in 2021, let's say you could have a saliva test for HPV. What do you do with that data? I think we don't know that yet, because it might be something you were exposed to last week that you're going to clear normally in 2 months. We don't know if we don't think that's probably putting you at a high risk of getting head and neck cancer down the road. But if you have chronic infection, one can imagine that we're going to eventually have saliva test available for clinical use. And if you have a chronic infection, maybe those patients are the ones that get the vaccine, and closer surveillance in the years ahead from a head and neck cancer standpoint. We don't have any screening for head and neck cancer beyond astute physicians and dentists looking for abnormalities. We don't have mammography or colonoscopies for head and neck cancer. We hope to someday have, saliva tests, serum tests, blood tests, but we're not there quite yet. There is now, as of just this spring, an FDA-approved test to test in the blood HPV DNA that is arising from a head and neck cancer patient. And there was an important article that came out almost a year ago that showed that if patients who went through primary treatment for their HPV head and neck cancer, if they have persistent HPV detected in their bloodstream, after they go through treatment, they're at high risk for disease recurrence. So maybe those are the patients that we can focus on to give more treatment after we thought we cleared them, if they have detectable HPV still around. And that's sort of finding that DNA of a tumor cell in their circulation, finding that needle in the haystack that says that they still have some cancer cells left in their body. So that's just being stood up now as a clinical test, but that's for patients who are already diagnosed with head and neck cancer, and we're using it for surveillance of their cancer. We don't yet have that being utilized as a surveillance, in the general population. So we've got a lot of work to go until we get to the point where everyone's vaccinated in the decades ahead. In the meantime, we got to turn this epidemic around and do better treatment for our patients. So the question is, do you regularly test for any tumor mutations besides P16? So great question. So the marker for HPV tumors is a protein called P16. It's sort of exclusive for HPV positive tumors. It's negative in HPV negative tumors. We usually stop there here at Inova. And when I was at Georgetown, we then did the next step to figure out, do the patients have HPV 16 versus 18 versus 31 and 35? That doesn't necessarily drive a treatment decision once you're either HPV or not. So we don't do the further genomic test, but one can. Low risk or the less frequent oncogenic HPV subtypes to really find out, are they 31, 35 positive and others. But we typically just stop at P16. Now the nuance to that is, so P16 is a protein that the pathologist can detect using immunohistochemistry, an antibody test that they do is in terms of a general pathology review. If you get a fine needle aspirate of a lymph node to diagnose squamous cell cancer, and you test that, so when you put a needle in, you pull out some cells, you put it on a microscope slide, oftentimes the P16 status of that can be falsely negative. So in our patients, if we have a oropharynx patient tumor that we got a lymph node biopsy and they came back P16 negative on the fine needle aspirate, we'll do another biopsy of the lymph node, a core needle biopsy or a tissue biopsy from the tonsil area to do the P16 stain, because it does drive prognosis and it's more and more driving treatment decisions, certainly trial enrollment right now. So the P16 is negative on a fine needle aspirate. Don't stop there. You might need to do a core needle to confirm whether that patient is HPV positive or not, because the fine needle aspirate is less sensitive to P16 testing, HPV testing, than a core needle or a primary tumor biopsy in the oral pharynx. So that's a great question.
Do we have any other questions? We have 2 minutes left. I guess before we go, if people want to learn more or follow the progress of your research, how can they do so?
Certainly at the Inova Cancer website, and our big meeting, our big cancer meeting every year. It's called ASCO, American Society of Clinical Oncology, is happening the first weekend of June. We're not presenting there, but other people are presenting there. We always, in the head and neck series, half of what we talk about is HPV positive cancer. I'm sure you'll see, news reports and things coming out either starting May 19th when the ASCO abstracts are presented, or it has to be released publicly, and then at the meeting itself, which is virtual this year, but will be, widely attended. 40,000 people from around the world get together to talk about cancer. I'm sure you'll see some news about HPV positive head and neck cancers and cervical cancer. That's where our treatment advances are released. So keep an eye out in that first weekend of June for more information coming out from researchers around the world, and hopefully, continuing to refine how we treat these tumors and ideally over time prevent them happening at all. So keep an eye out for that coming up in early June. So you might be on mute, Janae.
I'm just talking. Okay, thank you so much, Dr. Deeken. I really enjoyed it. I learned a lot, and I believe everyone else did as well. This is amazing. And if you're interested in learning more about oral health subcommittee that's to be coming, please just let us know in the chat or let us know via email. You can email us at VHIT@cancercoalitionofvirginia.org. We look forward to having you hopefully join us for our next session on June 2nd at noon. We will be talking about HPV and health equity with the VCU health team, oral health team. So we're excited for that. Please feel free to reach out to us via email. And thank you, everyone, for joining us. And that concludes this meeting. Let's see.
Thanks, everyone.